This video is about foundational concept number 1A, proteins and amino acids and their structures and functions. We're going to cover amino acid reactions, and what we need to know for the MCAT is sulfur linkages for cysteine, peptide linkages for polypeptides and proteins, and finally, hydrolysis. So sulfur linkages are created when two cysteine amino acids are lined up because they are both hydrophilic polar amino acids that create disulfide bonds or bridges by linking their sulfur atoms to each other. So when joined, the cysteines are collectively known as cysteine, and that is cysteine without an E. The MCAT will usually not test you on the difference in this nomenclature, but it is important in case it is part of one of the amino acid questions on your exam. It's important to note that this sulfur bond occurs in the side chains rather than the amino acid backbone, which consists of the alpha carbon, carboxylic acid, an amino group, and a hydrogen. When these two sulfurs join, they are oxidized and lose two hydrogen atoms in the process. Sulfur bridges are very important for helping stabilize tertiary and quaternary protein structures, what are going to be talked about in later videos. The next important amino acid reaction is the peptide linkage. Peptide linkage is a dehydration reaction as water, or H2O, is lost in the process. This creates peptide bonds, which are also known as amide bonds, which make up peptides, which comprise the primary structure of proteins. This interaction occurs between the amino and carboxylic acid groups of the amino acid. A peptide bond is an endergonic or energy-requiring process. This bond has partial double bond character, meaning it is difficult to rotate and can be considered a fixed bond. This occurs because the nitrogen has a lone pair of electrons which move between the bonds. This may seem counterintuitive that a peptide bond takes energy, but molecules enjoy being in a lower energy state for more stability. Ribosomes are a cell organelle responsible for making peptides and proteins. They catalyze the formation of peptide bonds while they read mRNA and translate amino acids to peptides. So, peptide bond formation is an example of nucleophilic substitution reaction. The nitrogen's lone pair attacks a carbonyl or CO group, forming an nitrogen-carbon bond. Electrons from the OH bond are sent to the carbonyl oxygen to reform a second bond, and the leaving group, OH, forms water by grabbing onto another hydrogen in the solution. Nucleophilic substitution reactions are very important to learn for the MCAT, so I'm going to identify the carbonyl group as an electrophile and the nitrogen as a nucleophile, and this will be talked about in more detail in a future video. So here's just a picture of the reaction summary. As you can see, the nitrogen from the amino group performs a nucleophilic attack on the carbon of the carboxyl group. So this forms an amide group, which is carbon attached to nitrogen, and the carbon is also bonded to an oxygen atom. The OH and hydrogen both leave together as water, or H2O. So peptide bonds are what make up the primary protein structure, as they are links between the amino acid backbone. They are always in carbon to nitrogen form, with the nitrogen terminus being open and a carbon terminus being open at the ends of the peptide. Often, peptides and proteins are thought of as being interchangeable terms. However, they are very different. So just have a quick summary here of the differences. So peptides are generally short chains of 2 to 50 amino acids that are connected via a peptide bond. They are well less defined structures and only are made up of primary structures. There's oligonucleotides, which are 2 to 20 amino acids, and polypeptides, which are up to 50 amino acids. For example, 2 amino acids makes up a dipeptide, and 3 amino acids makes up a tripeptide. When something is called a protein, it indicates that there are more than 50 amino acids, that there's long molecules made up of multiple peptide subunits, so polypeptides, can be digested by enzymes, which are other proteins, into shorter fragments, and it has complex conformations, including secondary, tertiary, and quaternary structures. We're going to be going into more detail on proteins in the next series of videos. Hydrolysis indicates that a bond is broken up with water. So if you break down the words, hydro stands for water and lysis stands for breaking. Peptide bonds are hydrolyzed when the peptide is in a strong base or there is an enzyme present. Hydrolysis is an exergonic process, meaning that it releases energy. The activation energy, so the energy that is needed to start this process, is high, making these peptide bonds quite stable with a slow breakdown. Hydrolysis is essentially the opposite of the peptide bond formation process and is going to be summarized on the next slide. This high activation energy is so important in order to make it difficult for proteins to just randomly break down within our bodies. So here's just a quick reaction summary. So as you can see, the lone pair on water's oxygen attacks the carbonyl carbon. The double bond electrons move onto the oxygen atom. The nitrogen leaves, and the newly formed amino group will be protonated by hydrogen in the solution. So we're left with the two newly formed segments, which look exactly like the amino acid backbone that we're used to.